this series. Order forms can be obtained from BBC Publications, Schools Orders Section, 144 Bermondsey Street, London SE1 3TEH. I'll just repeat that address. BBC Publications, Schools Orders Section, 144 Bermondsey Street, London SE1 3TH. And further details can be found in the leaflet sent to schools at the beginning of term. Now, science topic.
Just when you thought it was safe to go into the kitchen. This milk has been soured by microorganisms, and microorganisms are all around us. Though they grow best where there's plenty of moisture and food. Molds are the most obvious microorganism. We can see them when they've grown on food. They may have spoiled this cheese, but they're not always a nuisance. It's blue mold that gives this cheese its distinctive flavour. One set of microorganisms you won't normally see are bacteria. They multiply fast in the kitchen. This dishcloth is full of them, as we can see if we look at just one fibre through a microscope. The bacteria grow well in the washing up water with all the food from the dirty plates. Some bacteria can double in number every 20 minutes. There won't be time for that in the washing up bowl, but some of the water tipped down the sink may stay in the U-bend for hours. The bacteria won't do much harm there, but they can contaminate food. And of course, a lot of food and bacteria pass through here. These are mainly human gut bacteria, and too many of them can also be dangerous. Fortunately, they don't stay long. Flushing is a good disinfectant. They end up here at a sewage treatment works with lots of other pollution. The job of the sewage works is to get rid of the pollution, and this can be done by putting some of the microorganisms to work in controlled conditions. This is Beckton Sewage Treatment Works, which every day processes over 1 million cubic metres of sewage from 2.5 million Londoners. It's the largest sewage works in Britain. So how long has sewage been treated scientifically?
We've been using biotechnology since the 1930s here. The scientific research showed that you could actually concentrate the naturally occurring microorganisms in sewage and use those organisms to treat the pollution in the sewage. The site here at Beckton treats such a large volume that it really is a major civil engineering job, as well as using the biological process. So it's a combination of both technology and biology. The process really starts with primary sedimentation. The primary sedimentation tanks are used to settle by gravity the particles in the sewage, and this removes about 50% of the pollution load in the sewage.
The pollution settles as sludge on the bottom of the tank. Each tank has a large scraper that sweeps the sludge forward into a hopper, and it's sent on for further treatment. The remaining liquid can go on to the biological aeration tanks. Here it's the aerobic microorganisms in the sewage that are put to work. They're mainly soil organisms, which use the pollution as their food, but they need a lot of oxygen to help them break down such large quantities. They take their oxygen from the air that's pumped into the tank through hundreds of rows of air diffusers. You can see the rings of air bubbles on the surface of the liquid. By supplying air to the microorganisms, large amounts of sewage can be treated in a few hours, quantities that would take microorganisms in the river weeks or months to deal with. The air in the biological aeration tanks keeps the microorganisms well mixed up with their food. The microorganisms come in lots of different shapes and sizes. The microorganisms use the pollution in the sewage as their food. By the time they've done their work, they've broken the pollution down into harmless substances like water, carbon dioxide and nitrates. From the aeration tanks, the sewage goes to the final sedimentation tanks, where the biologically active sludge is settled out by gravity and recycled. These samples show how the pollution has been reduced from the raw, untreated sewage to the final clear liquid. But what happens to the raw sludge from the beginning of the process?
The thickened sludge from the primary sedimentation tanks is further thickened by gravity again before being fed into the sludge digestion tanks themselves. And in those tanks is another biological culture. This time it's an extension of the human gut bacteria that work in the absence of air. These are called anaerobic bacteria, and they have to be maintained at between 30 and 35 degrees centigrade to work efficiently. These take the raw sludge that we feed them in as a food supply, and they will gradually break them down and produce as end products carbon dioxide and methane gas. And we use the sludge gas produced to power gas turbine engines, and that produces some of the energy that the works requires. At the end of the process, we end up with 6,000 tonnes of digested sludge every day, which, because it's been biologically treated, is loaded onto our ships and disposed of to sea. The liquid portion, about 1 million cubic metres every day, is discharged directly into the River Thames with 97% of its pollution load removed.
Sewage has been thoroughly treated for only a few decades, but biotechnology has been used in food processes for centuries, though without people knowing how it worked. A lot of biotechnology is based on fermentation. In the case of beer, the microorganism used is yeast, which is fermented to produce alcohol. Yeast can also make bread rise. This is a special strain of mould used in blue Stilton cheese. And bacteria like this produce lactic acid, which starts the whole cheese-making process off and affects the body and texture of the final product. How has biotechnology helped with a traditional cheese like blue Stilton?
Well, basically it's helped us to eliminate the large elements of chance which were present in cheese-making of old. It makes us more efficient, and it makes our operations far more cost-effective. Today we can make up to approaching 58,000 litres of milk a day into Stilton cheese. When the milk is actually in the vats, we add to the milk a small quantity of starter. Starter is a culture of lactic acid producing organisms, and this increases the acidic development of the milk throughout the process. It plays an essential role in ensuring that the right body and texture are obtained in the cheese.
Because the cheese is blue Stilton, a special culture of blue mould is added, though because the mould is aerobic, the spores will stay dormant in the milk until later in the process. The next stage is to add rennet. Rennet is a biological enzyme which coagulates the milk. After 40 minutes, the curd is tested by hand to see if it's set. It should split in a clean, straight line. That means it's ready to be cut. It's hard work cutting a 4,500 litre blancmange, but it's essential to make the solid curd separate from the liquid whey. The curds and whey are then run off into another vat called a cooler, where the whey will be allowed to drain off overnight, leaving the curds behind. The whole process is very carefully controlled to provide the best environment for the microorganisms to work in. In the factory laboratory, a number of tests are carried out on the raw milk and the whey. And tests are also done on the starter culture to make sure the microorganisms are active enough to produce a good quality cheese. The next day, when the whey has drained off from the coolers, the curds go on to the next stage of the process. They go through a cheese mill, which breaks the curd into small pieces. The curd is raked and salted. After further raking, the curds are put into special cheese-shaped containers called hoops. The new cheeses are stored in the hoop room for a few days. They're turned regularly, which compresses the cheese slightly, but not too much, as the aim is an open texture to help the mould grow. But the mould mustn't grow yet, so the surface is sealed in the binding room. The soft cheese is spread into the gaps to make a smooth, airtight coating. The cheese is then stored for several weeks to mature, and its rough, characteristic coat develops. But what about the dormant mould spores inside the cheese?
At about 8 weeks, when we decide the cheese curd is developed to the right stage, we then wish to promote the development of blue mould within the cheese. Now, to facilitate this, we have to allow oxygen to get into the center of the cheese. So the next process, which we're involved with, is actually piercing the cheese with a machine, which we call a piercer, which simply involves placing a series of stainless steel needles into the center of the cheese. So as soon as you pierce the cheese, the oxygen activates the mold spores, and we start to get the development of blue veining within the cheese.
The factory employs cheese graders to check the development of the cheese before it's sold. Modern cheese making is now a well-controlled scientific process, so rejects are rare. It's a successful combination of modern technology and traditional methods. Each cheese is individually checked by hand. There are 100,000 cheeses in this store. 65 cheeses can be made from each of the 4,500 litre vats of milk, and the factory makes over 250,000 cheeses a year. Many of the cheeses are sold whole, but some are cut up in wedges and then packaged ready for the supermarket shelf. Apart from food, perhaps biotechnology's most important contribution is to healthcare. There are millions of doses of vaccine in this storeroom at the Wellcome Research Laboratories. Some bacteria cause disease, but we can use them as microbiological factories to produce vaccines that will prevent the disease. The bacteria have to be very carefully controlled. The technology is now available to grow hundreds of litres of bacteria in large fermenters. The process is constantly monitored. This is an aerobic fermentation suite, so the oxygen level is controlled as well as the pH and the temperature. Sterile protective clothing is needed in this diphtheria suite because the toxins the bacteria produce have to be treated with great care. There are anaerobic fermenters as well. This one has a capacity of 1,000 litres and contains a culture of bacteria for an animal vaccine. But before this commercial scale of production is reached, a lot of research has to be done on much smaller quantities. What does the laboratory work involve?
The two basic elements are the biology, of the work, growing up the bacteria, and the technology of taking it up to production. You've got to have the two know-how before you can get a product to the marketplace. You've got to be able to grow your bacteria to start with, and once you are able to grow it, then you've got to be able to scale up and have the technology to grow it in bulk and to harvest it and to purify it.
A new purification process has been developed in this lab. It's only after starting with quantities this small and gradually scaling up the process that you can overcome the problems of dealing with commercial production quantities, like those produced by this fermenter, which has a capacity of 1,500 litres. The production of vaccines and all the processes we've looked at so far exploit naturally occurring microorganisms. But now we can go one step further. These bacteria are a laboratory strain of the human gut bacteria E. coli, and they've been genetically engineered to make them produce something rather unusual, human insulin. It's a very complicated process, but it's recently achieved full-scale commercial production. It means we now have an alternative to animal insulin. We've looked at the products of biotechnology, but what about the consumer? Sue Callar is an illustrator. She's also a diabetic. Her life depends on her daily injections of insulin. When she was 25, Sue's pancreas stopped producing insulin. Her body still converts food to glucose, but without insulin, she can't use it for energy. When tests confirmed that Sue had diabetes, she was told she'd have to inject insulin. How did that affect her life?
Well, I must say, first of all, it did turn my life upside down, of course. I mean, being told that you have to inject for the rest of your life isn't a very nice thing to have to be told. But nevertheless, once you get into a routine and work out your eating regime, it's fine.
So what is the daily routine for diabetics?
They have to be very careful of their diet, and they have to eat regularly and at regular times, because the insulin works on a time basis, so that if I've taken the insulin, I can't decide to eat and then not eat later on. I have to eat at regular intervals. And it can be just as dangerous to have too much insulin as not have enough.
What sort of insulin does Sue use?
I use human insulin, which is fantastic because it's genetically engineered and identical to human insulin. So, personally, I'm a vegetarian, so it's great for me that it's not an animal product like the other insulins.
Also, there's a limited number of animal pancreases available for processing, but humulin can be produced on demand for the increasing number of diabetics, 2% of Britain's population, and over 60 million people worldwide. How much insulin does Sue use each day?
I take 3 injections daily. I take one in the morning, one at 6 o'clock at night, and one at 10 o'clock. And the 10 o'clock one takes me over through the night, so that I always have a supply of insulin in my body.
Sue has her diabetes well under control, and she recently had a very healthy baby. So for Sue, production of human insulin on a commercial scale is a major success story. Biotechnology has made a big difference to Sue's life. So how does she feel about it?
Well, I think it's absolutely fantastic that the research has produced this insulin, because otherwise I and thousands of others simply wouldn't be around. And it enables me to lead a normal life.